Welcome to the GCSE Biology tutorial for topic B2 organisation. This tutorial will look at plant organisation. Starting off with, you need to know that plants are made of organs as well, just like animals, and these organs are things like your stem and your root, your leaves, your flower, and organs are made of tissues, so groups of cells working together. For example, the epidermal tissue or palisade tissues. We'll look at more of those later. First of all, you need to know what happens inside the leaf. So here, if you take a leaf like this and you zoom into the side of it, these are different layers and the different tissues. You've got the waxy cuticle on top, and that reduces water loss by evaporation, so prevents water evaporating from the top. The upper epidermis is also transparent, so light can go through. You've then got the palisade mesophyll layer, and this has got lots of chloroplasts inside it to trap sunlight for photosynthesis. You've then got a spongy layer, which has got air spaces to allow for diffusion. You also have different vessels inside a plant leaf. You have the xylem vessel, which delivers water, and the phloem vessels, which transport glucose. The lower epidermis has got holes in it, and these holes are called stomata, and they allow carbon dioxide to move into the leaf and oxygen and water vapour to move out of the leaf. And stomata are controlled by guard cells. So this is what they would look like under a microscope. So these are the guard cells and the holes are the stomata. So when the cell has been watered a lot, has been given lots of water, the guard cells get bigger and fill up and they become turgid. And that makes the stomata open, so that makes the gases exchange. And when it hasn't got enough water, then the opposite happens and the guard cells become flaccid and they close the stomata, and that stops too much water leaving. So this control prevents too much water leaving the plant. It also, the guard cells are sensitive to light, so they would open at the day and close at night for photosynthesis. Stomata are found underneath the bottom of the leaf. There is some leaves that have them at the top, for example lily pads, because their bottom is on the water. Let's look at those vessels. So just like arteries and veins in a human, a plant has vessels to transport substances. One is called xylem. So xylem tubes are made of dead cells and they're just hollow tubes. And they're made stronger by a chemical called lignin. And xylem carries water and minerals from the roots all the way up to the leaf and the stem. And they allow for this process called transpiration, which we'll talk about later. Phloem tubes, they're made of living cells and they've got little holes in them at the end of each one, which connects to other cells called companion cells and sieve cells. They transport glucose that's made from photosynthesis, and they transport it around the plant for use and for storage. The movement of glucose is called translocation. So let's look at transpiration again. So transpiration is the movement of water through a plant. Now to understand this, we need to go into a bit more detail. When water leaves a plant by evaporation in the stomata, it comes out of the holes. As it draws out of the holes, it causes a shortage of water in the leaf. So the water then draws up more water from the rest of the plant. That leads to water being drawn up from the roots, and this creates a constant stream of water moving through the plant called transpiration stream. So in other words, the suction and evaporation of water at the leaves makes more water sucked up at the roots. There's certain things that can affect transpiration. So light intensity, when it's brighter, it means the stomata are open, so that means there's going to be more evaporation, more transpiration. When it's hotter, there's going to be more evaporation, more transpiration. When it's windier, then there's more that water vapour will be swept away from the stomata, so there's more transpiration. When it's more humid, there'll be less transpiration. So drier air is when there's more transpiration. You can measure transpiration using a potometer as shown here. And the water uptake by the plant is linked to the water loss. So how much water it's sucking up, you could estimate that's the same as how much water it's losing. And you would set this plant up with a capillary tube to make a bubble in the water and then measure the distance that bubble travels. And the distance it moves per unit of time can be used to determine how much water is being lost from the plant. You can do different things with this experiment. You can change the light intensity or change the temperature and see if that affects how quickly that water bubble moves. So, there's a 6-mark question here for describing how leaves are adapted to their function. This is a popular question, so pause the tutorial, give yourself 10 minutes. So, how is a leaf adapted? First of all, leaves have got a large surface area to absorb as much sunlight as possible for photosynthesis. Starting with the top layer, it's a waxy cuticle that prevents the water evaporating from the top. The top layer is also transparent to allow light to go through. You've then got a layer of palisade cells, and they are full of chloroplasts to trap sunlight for photosynthesis. You then have an area called the spongy mesophyll. This has got air spaces, and so gas exchange can happen here with carbon dioxide and oxygen. At the bottom of the leaf, you have the stomata, which are the holes, controlled by guard cells, and that allows gas exchange, so it allows carbon dioxide to enter the plant, and it allows oxygen and water to leave the plant and allows for transpiration. There's also vessels in the leaves, so the phloem, which transport glucose, and the xylem, which transport water. We've looked at plant organisation and transpiration.